Today, we invited 7 speakers from the NIH Brain Initiative Cell Census Network, BICCN, to present their recent work using scalable single-cell approaches to characterizing brain cell diversity. Their research is a major effort of the BICCN, founded about 2.5 years ago. The overarching goals are to generate comprehensive as well as highly granular reference brain atlases that will integrate molecular, anatomical, and functional features of brain cells, and to develop new tools for experimental access to the different cell types in alignment with the #1 priority of the Brain Initiative as recommended in Brain 2025 report. This symposium, entitled Molecular Taxonomies of Brain Cells, will showcase several BICCN projects from identifying brain cell diversity, establishing brain cell taxonomies, to developing new cell type specific targeting tools in a manner not requiring germline genome modification. The symposium will present first a comprehensive single-cell transcriptomic survey and identification of new cell types in mouse cerebellum by Evan McCusker. Second, cross-species evolutionary study of brain cell types in marmoset, human, mouse, and other species by Fenner Queenan and Trev Bakken. Third, comprehensive epigenomics characterization of brain cell types by Sebastian Crystal and Zhu Zhang. Fourth, data integration and cross-validation by Iran Mokamel. And last, single-cell multi-omics analysis and the new viral tools targeting brain cell types by Bosilka Tazik. Now, without further ado, let me introduce our first speaker, Evan McCusker from Broad Institute. He's going to talk about, his title is A Transcriptomic Atlas of the Mouse Cerebellum, Reviews Regional Specialization and the Novel Cell Types.
Thank you very much, Yong. As Yong mentioned, I will be telling you today about a comprehensive cell atlas of the mouse cerebellum, which is part of a larger atlas that we are constructing using droplet-based single-nucleus RNA-seq across the entire mouse brain. And this is the first portion of the data set that has been completed and analyzed. The data set was constructed by taking a total of 6 mice, 4 male, 2 female, separately dissecting each lobule of the cerebellum, processing it through a newly optimized form of single-nucleus RNA-seq, which yields very high numbers of genes per profile, to yield a total of 800,000 profiles across all of these individual 16 lobule regions. We then performed Liger, an analysis tool that we developed previously, and a total of 611,000 quality-controlled profiles distributed across both glial and neuronal types. Here is an image of the overall dendrogram of relationships amongst the 46 clusters we identified. On the top are glial clusters, which were largely shared with other brain regions, except for the very unique Bergman cells. And then on the bottom are a series of neuronal types that we identified that are quite specific to the cerebellum. Using the Allen Brain Atlas, we were able to annotate the locations and the identities from previous work of the major neuronal classes, the Purkinje cells, granular cells, and then Golgi interneurons and a variety of other interneurons. We used this data set to answer 2 big questions in the cerebellum. The first big question was, how much regional variation is there across individual cell types? And this arises from a larger question that's been in the field for a while in the cerebellum, which is to what extent are the circuits of the cerebellum, which is a very large structure, the same across all these lobules versus different? And the canonical view is that they're relatively uniform, that input comes through the climbing fibers, and that output is through the Purkinje cells. So the large scale circuit is is sort of thought to be the same across the individual lobules. But there are some hints of alternate ideas of this, and the most commonly cited one is this zebrin pattern of expression in the Purkinje cells, that there are certain genes that seem to be longitudinally expressed across these zebra-looking stripes in the cerebellar structure. Indeed, when we looked at our Purkinje profiles, we found that there was ALDOC positive and negative subclusters within our Purkinje analysis, and that there was a much greater specialization amongst the Purkinje clusters that were ALDOC positive than ALDOC negative. And furthermore, that the ALDOC, the populations that we identified, these 9 different Purkinje populations, seem to be particularly regionally enriched in posterior lobules, suggesting that the posterior lobules are considerably more diverse in their cell specializations than the anterior lobules. Another big question that we had, of course, whenever you're doing one of these large-scale surveys of a tissue, is, are there any new cell types that we didn't know about already? And indeed, there was a population that seemed unique to us and one that was hard for us to annotate. And they were in the molecular layer of the cerebellum. Now, in the molecular layer, it's been known that there is a continuum of morphologies that exist, from the outer third, where stellate morphology cells exist, and then the inner third, where these basket cells exist. And these morphologies were initially identified by Cajal more than 100 years ago. In our taxonomy, we identified 2 populations, which we call MLI1 and MLI2, which localized to the molecular layer. We did initially confirm this using our technology SlideSeq, but we also confirmed it, as I'll show in a minute, with single molecule FISH. And the MLI1 and MLI2s are quite distinct. They have many, many different markers that separate them. And in fact, the MLI2s look more like Purkinje layer interneurons than molecular layer interneurons. And here is a FISH image to show you that there is this pair of populations separated by these genes SORX3 and NXPH1, both present in the molecular layer, about a 2-to-1 ratio of these 2 populations. We were curious to know what these populations actually did in the cerebellum, so we developed a pipeline so that we could jointly molecularly phenotype, physiologically phenotype, and morphologically phenotype these cells. The way we did it is we patched cells in acute slices. We filled the cells with an Alexa dye, and then we did post-hoc HCR, single molecule FISH, to identify which of these 2 MLI populations there were. The long and the short of it is that the 2 populations both show a continuum of basket and stellate morphologies, which is very surprising to us. The same continuum from the distal part on the top, where both MLI1s and MLI2s look stellate-like, to the inner portion of the layer where they both looked more basket-like. But nonetheless, they had very, very distinct physiologies. They were different in their firing rates, in their excitabilities, and then most strikingly, which I'll end with, is they actually have differential gap junctioning properties. And this was first hypothesized from our data because only one population, the MLI1 population, expresses the canonical gap junction gene connexin 36. Which we confirmed by single molecule FISH. It's extremely unique to MLI1s compared to MLI2s. And then our collaborators in Wade Regeer's lab recorded from individual cells using a technique that allows them to identify electrical coupling, these spikelet recordings. And they found only spikelets to be present in the MLI1s and not in the MLI2s. So in conclusion, we've constructed a data set that provides a community resource to basically identify all the molecular phenotypes of cell types within the cerebellum at lobular resolution, so we can see the differences across each individual region. We see more molecular diversity in the posterior lobules. This is exemplified by the Purkinje cells, but it's also the case for the granular cells and the other cell types where we also saw some regional specialization. And we also identified a new cell type that's extremely distinct from the molecular layer, two sort of populations within the molecular layer that are quite distinct, despite the fact that they have nearly indecipherable morphologies across the tissue layer. So with that, I would like to just thank our collaborators in the Wade Regeer's lab and also in Aviv Regev's lab, and also recognize the folks who led this work, especially Valina Kozareva and Carly Martin. Thank you very much. So now I'd like to introduce Fenna Kreenan from Harvard Medical School.
Hi, everyone. Thanks, Evan, for the introduction. Thanks to the organizers and to Yong for moderating this fascinating session. So I wanted to share some of the work that was really enabled by our participation in the BICCN. So we've been generating a single cell molecular and cellular atlas of the marmoset brain. And one of the first things we wanted to ask or evaluate and that you might wonder is what really does a primate buy you in terms of conservation of the cell types and molecular programs that we might see in humans. And so to initiate these kinds of questions, we took a comparative approach, and we really focused on interneurons in particular, in part because previous work looking at molecular taxonomies of cell types has emphasized, and it's true at the broadest level, they are quite conserved relative to other neuronal types, all the way from reptiles to humans. And also because we and others have seen that the molecular profiles of specific subtypes of interneurons, at least in the neocortex of mice, tend to be quite stable across areas. And so literally interneurons made quite a conservative but then potentially informative place to start these kinds of comparative questions. And we acquired data from 3 primates, from humans, macaques, and marmosets, as well as from mice and ferrets, sampling multiple neocortical regions, the striatum, hippocampus. And of these larger data sets, we carried forward about 180,000 interneurons for our analyses. And when we began this project, we started with really a simple list of the ways we might expect brain cell types to evolve. Actually, we only thought of 3 of these, but found a 4th in the process of this work. So we thought you can change the abundance of a conserved type across species. You might change the genetic program or molecular details of a conserved type. You could allocate the same type differently across brain structures or contexts. And then potentially you might also invent a novel type. So to the first point, consistent with stereology, we found that interneurons, as a percentage of all neurons in sensory cortical areas like Z1, made up about 13 to 20% of all neurons, and that's quite consistent with the literature. And is also quite stable across species. Primates, though, allocate a far greater proportion of interneurons to the higher order association areas like prefrontal cortex, but also higher order areas in parietal and temporal lobe, and relative to mice, who tend to have more uniform distribution. What we didn't expect was that the ferrets actually allocate interneurons in proportions that resemble primates much more than they do mice. And so in this regard, and contrary to what you might predict by evolutionary divergences of these species, the mice appear to be the outlier. We can also, of course, examine relative proportions of subtypes of interneurons, such as those arising from different embryonic origins. And this all, again, reinforces to us that primates seem to customize the allocation of these major types within different neocortical areas, and that might contribute to neocortical specialization. And again, this is important for us to see that a marmoset, even though it has a small and smooth neocortex and diverged from us over 45 million years ago, seems to allocate interneurons in these basic proportions in very similar ways to humans. And turning to another way that cell types might evolve, we found that neocortical neurons in all 5 species could be partitioned into 4 main categories. And they can be nicely marked by canonical markers like parvalbumin, somatostatin, and so forth. And we could then examine the relative expression levels of other genes within those main conserved classes. And this plot on the right really illustrates one of the main patterns, which is that energy expression, so it's one such gene that has quantitative variation amongst interneuron types. Energy expression levels in humans can be quite nicely predicted by the relative levels of the gene in marmoset or macaque interneurons, but not so much so by the levels in the non-primates. And so we can summarize this behavior broadly across hundreds of genes that are expressed in all 5 species within interneurons in cortex, and show that evolution really constrains the quantitative expression levels of genes, even if they are pan-neuronal, that is, not marker genes for any particular population. I want to briefly foray into another facet of this idea of changing the molecular composition of conserved types. Because remember, I told you that both we and others have observed that interneurons in mice really don't vary that much by cortical area, relative at least to excitatory neurons, of a given flavor of interneuron, that is. But because we had collected so many neocortical regions in the marmoset, we could ask this question in a primate context. And it turns out that the same subtype of interneurons, say a parvalbumin subtype, sampled across multiple cortical regions, does have quite a lot of variation in gene expression, and this variation seems to follow an unexpected spatial logic. That is, if you line up the cortical areas from the anterior pole to the posterior pole, you can find dozens, if not hundreds, of genes whose expression level correlates with that spatial arrangement. We were able to validated some of these with single molecule fish, such as here where you see the gene ASS1 in purple is more expressed within parvalbumin interneuron types in the frontal portions of the marmoset neocortex relative to the parvalbumin interneurons in the back. And this could be quantified from the single molecule fish images. So it turns out that interneurons may retain their spatial positioning information within the quantitative deployment of many genes. And what's interesting is that these same genes actually exhibit the same regional bias within the other species as well. Of course, the magnitude and the percent of agreement is higher within the primates, but even the non-primates seem to follow this kind of spatial logic. So the trend is there for all of them, supporting this idea that maybe interneurons encode some aspect of their regional position within the neocortex. Turning to the third possibility, so just briefly, we observed what is a special case of modifying abundances, and that is that there's an interneuron subtype that is abundant in the mouse hippocampus, also in ferret, but has become rather abundant in the neocortex of primates as well. And this, of course, raises really interesting developmental questions about how interneurons get allocated across structures. And I should mention that Gord Fischel, who was a collaborator on this project, had this initial insight of looking across structures to unify molecular types. And then finally, what we expected that if anything, if we're going to observe a novel type, that it would be in the neocortex of the primate. Actually, we were surprised to find a novel type rather in the marmoset striatum that we didn't observe in mouse. And this is a type that expressed a unique suite of neuropeptides like TAC3 and VIP, but also transcription factors like LHX6, suggesting that it may have an MGE origin like most of the striatal interneurons. What was interesting, though, is that this type we could also observe in humans, but not in ferrets, suggesting that it might be an evolutionary gain after the primate-rodent split. So with that, I just want to close thanking my collaborators and just suggesting that I think we and others will continue to find many more such examples of evolutionary novelties in conservation and divergence as we survey more regions and species. And that is the perfect segue to introduce Trygve Bakken from the Allen Institute, who will be discussing work on evolution of cellular diversity in motor cortex of human, marmoset, and mouse. Thanks.
Thanks, Fenna, and thanks, Yong, for organizing this session. Today, I'd like to tell you about work that was really done in collaboration with many of the BICCN teams, and this was to look at a functionally conserved area, primary motor cortex, across primarily 3 species, human, marmoset, and mouse, with a small subset looking at macaque layer 5. And this collaboration extended both to the data generation and on the analysis side, and acknowledging some of the groups on the right there involved. The different modalities that we were profiling cellular diversity in this area included 2 RNA-seq assays, one for full-length transcripts, SMART-seq, and a droplet-based method from 10x Genomics, DNA methylation, and open chromatin, either alone or in conjunction with RNA from the same nuclei. So these assays were all profiling single nuclei in order to increase our ability to compare across species. And I'm showing on the bottom an example of dissociating these nuclei from postmortem human tissue, where we're able to identify in coronal slabs the region of interest based on gyral folding patterns confirmed based on the histology, particularly these large pyramidal neurons in layer 5 called BET cells, and then dissociated single nuclei and enriched for neurons by sorting for nu n, and then profiled with multi-omics methods listed above, and then performed unsupervised clustering on each individual modality. So this effort resulted in approximately 100 cell types identified in each of the species that have been arranged in taxonomies on the left. And really the highest resolution taxonomy came from the droplet-based RNA-seq method, and that's what's shown here. We could then leverage shared co-expression across these species to integrate nuclei into a common space, and then perform unsupervised clustering in that space to define a final set of 45 consensus types that were shared across these species. There were a couple of types that were unique to species that I won't address here, but just to say that there are some, for example, progenitor populations of glial cells that are more found in mouse than in primates. But these consensus types included a number of types that matched one-to-one across species, including chandelier cells. Although of note, there is a very rare subtype of mouse chandelier cell that we didn't see in the primates. But then some many-to-many matches, such as basket cells. We also, of course, had the epigenetic information for these different species. And what we found is that the gene regulatory signatures were highly consistent with expression. So here I'm showing on the left an integration of human glutamatergic neurons profiled with RNA and DNA methylation, and have been integrated into this space based on hypomethylation of gene bodies and marker gene expression. And sort of an unsupervised clustering, independent of the RNA clustering, using the DNA methylation data, could then be mapped back to the RNA clusters based on correlated marker expression. And we found many one-to-one matches, and also some one-to-many matches, likely due to less sampling of nuclei in the DNA methylation data set. So with this epigenetic, we could also do a similar process for the open chromatin, where we also found very good alignment. And then with this epigenetic data in hand, we could ask questions such as, are there conserved transcription factor binding site motifs across species? And this is, in fact, what we found. So I'm showing an example here for LAMP5 interneurons, a subclass of interneurons, where each row on the left shows enrichment for different transcription factors, both in the hypomethylated regions and open chromatin for this subclass, and corresponding RNA expression for that transcription factor. And these were quite conserved across the species, but distinct between subclasses shown on the right. So this is a TSNE showing in colors the different excitatory and inhibitory subclasses, and in different shapes of point are the different species. We could also ask questions like to try to relate conserved cellular properties to conserved molecular properties. So chandelier cells have been well described in a number of species and share common distinct features relative to other fast-spiking interneurons, the basket cells, in human and non-human primate and mouse shown on the top. And likewise, we could find a set of 25 marker genes that discriminate chandelier cells from basket cells and were shared across the species. And these genes included 3 transcription factors, including this gene ROR-A, which in a similar analysis to what I showed previously, we showed significant enrichment in chandelier cells compared to basket cells, the motif binding site for that transcription factor, both based on the DNA hypomethylation and the open chromatin. Finally, in addition to many conserved genes, as Fenna mentioned, there are many genes with differential expression across species for homologous types. So here I'm showing just the GABAergic neurons in a two-dimensional projection of the integrated space, a UMAP projection, where the colors indicate the species are well intermixed, and the different colored islands correspond to the major subclasses of GABAergic interneurons. And we can identify on the order of 1 to 200 markers within each species for each of these subclasses, including parvalbumin positive interneurons. But only a small subset of those are shared across all three species, with more sharing between primates than with mouse. And that's indicated both by this Venn diagram and also on the right for each of the subclasses, showing a limited set of conserved markers and many species-specific markers. So what I think I've shown is that there are a conserved set of types that have conserved for at least 65 million years and therefore likely point to a functional significance of those types. There's putative developmental mechanisms for cell type identity suggested by this conserved transcription factor code, and there is evidence for specialization of types across species based on divergent marker expression. So with that, I'd like to thank not only all the BICCN collaborators who contributed to this work, but the various teams that contributed from the Allen Institute. And of course, I'd like to thank our founder, Paul Allen, for his vision and support. And I'd like to introduce our next speaker, Sebastian Preissel from UC San Diego.
Thank you, Trygve, and thank you very much to the organizers for giving me the opportunity to talk today about our study on single-cell chromatin accessibility landscapes of mouse brain cell types. And the overarching question that we're trying to ask is, what are the gene regulatory programs and genomic elements that are underlying the unique identity and function of brain cell types? And we're really trying to understand the diverse molecular cell types identified by high-throughput single-cell RNA-seq analysis and spatial transcriptomics, how these cell type-specific gene expression patterns established and how are they encoded in the genome. And therefore, we are striving to map the cis-regulatory elements, in short CREs, in the mouse genome in brain cell types. And these cis-regulatory elements, which can be separated into enhancers and promoter elements, are characterized by and can be identified by open chromatin, which can be targeted by the method ATAC-seq. And in order to profile open chromatin regions in specific types in the brain, we employed a highly scalable single-cell ATAC-seq method, which uses a combinatorial bulk coding split pool approach, where we in the first step perform fragmentation in 96 well plates with the well-specific barcode, pull all nuclei together and separate them into a second set of 96 well plates with the indexed PCR. Using this highly scalable approach, we were able to generate single nucleus ATAC-seq profiles for more than 800,000 nuclei from 45 dissected brain regions, which were dissected from coronal sections along the anterior-posterior axis of the mouse brain, and were part of 4 main regions: the isocortex, the olfactory bulb, the hippocampus, and cerebral nuclei, which comprise striatum and pallidum. Using this 800,000 nuclei, Dr. Yang Li and Bing Ren's lab performed iterative clustering to identify not only the cell classes, non-neuronal, GABAergic, and glutamatergic neuron, but also 43 major neuronal types, and could further subdivide them into 160 subtypes. For cluster annotation, we used chromatin accessibility at promoter and gene body regions of canonical marker genes, and could see that, for example, in the major types, we found D1, D2, medium spiny neurons, hippocampal, granular cells, and several layer-specific cortical glutamatergic neurons. We could then, using the single-cell ATAC-seq data, we could further subdivide these major types into subtypes. And for example, I'm showing you here for the SST inhibitory neuron subclass, we actually identified 10 subtypes, and one of these represented the long-projecting GABAergic neurons indicated by accessibility at the chondrolectin locus. We next asked, how does this molecular taxonomy from chromatin accessibility compare to taxonomy defined by single-cell transcriptomes, and for that, we integrated our single-cell attack-seq data with single-cell RNA-seq data from Stan Dinnerson's group for matched brain regions, and could identify that for 160 single-cell attack-seq clusters, we found a match in the single-cell RNA-seq taxonomy, which indicates that there's a well correspondence between both taxonomies. And motivated by that, we were wondering, can we now go the next step to identify specific regulators of this cell type specific gene expression? And here I'm showing you our strategy to identify putative enhancers for cell type specific target genes. First, we identified these elements, these distal elements, out of our catalog of 490,000 elements which are co-accessible with promoter regions, which indicates that they might likely regulate this gene or could regulate this gene. And in the second step, we performed Pearson correlation between accessibility at this distal cis-regulatory element and RNA expression in the joint clusters. Using this approach, we were able to identify 130,000 positively correlated pairs, which comprised roughly 86,000 putative enhancers, which were linked to 10,000 genes that were expressed in the brain cell types. So on average, we identified 7 enhancers for any given gene promoter, and the average distance between enhancer and gene was around 180 megabases. Next, we were wondering if using this comprehensive list of putative enhancers, if we could get insight in what are the enhancers in specific cell types. And to do that, we grouped the enhancers into modules based on accessibility across the joint clusters, and could identify 38 modules, with one on top a module which was broadly accessible across all neuronal cell types. And all the other modules, all the other 37 modules showed a more cell type restricted accessibility. Notably, this accessibility was highly correlated with and highly mirrored the target gene expression. And notably, we didn't see such high variation in accessibility at the promoter regions, which further highlights the notion that enhancer regions really drive cell type specific gene expression patterns. And we further wanted to know what are the transcriptional regulators driving this accessibility. And for example, when we look at modules that were specific to microglia, we found enrichment for PU1A factors, for cortical glutamatergic neurons, strong enrichment for BHLH, NurD, NurG family factors. But we were particularly interested in this module 1, the top module, which seemed to be accessible across all neuronal cell types and has lower accessibility and expression in non-neuronal cell types. And surprisingly, we found a strong enrichment for CTCF, MEF2D, and RFX factors in this module. And it's particularly interesting with CTCF. CTCF is a ubiquitously expressed DNA binding protein which well described functioning as an insulator and chromatin organization involved protein. And it was just recently shown that it can also be involved in neurogenesis by regulating the protocaterin locus. But our data here actually show that it's involved in a much higher variety and many more biological processes which are relevant to the neurons, including axon guidance, synaptic transmission, and so forth. And I'm just showing you here one example gene, LGI1, which is involved in regulating presynaptic transmission. And in this locus, we found a distal element with CTCF binding site that interacted with the promoter of LGI1 in a specific way in neuronal cell types, but was not accessible in the non-neuronal cell types. And this accessibility was also accompanied by a low DNA methylation value, further strengthening the notion that these are actually cis-regulatory elements driving expression for LGI1. And we have also set up a web portal for exploration of the data, so please feel free to check it out. And with that, I would like to sum up by concluding that we have generated, I think, a very large community resource of 800,000 nuclear chromatin accessibility profiles, which corresponded roughly to 160 subtypes. We've generated a candidate cis-regulatory element list of 490,000 elements. subset of these, 85,000 could be linked to roughly 10,000 putative target genes. And finally, we indicate a role of MEF2, RFX, CTCF factors in regulating pan-neuronal enhancers and regulating a broad spectrum of neuronal processes. And with that, I would like to thank our collaborators, and especially Margarita Behren's lab, and Margarita Behrens and Jacinta Lucero for making the dissections and preparing the nuclei, Xiaomeng Hu and Xinqin Wang for generation of the data, and Dr. Yang Li for data analysis. And with that, I would like to close and hand over to Xuzhu Zhang from the Salk Institute.
Thank you for the introduction from Sebastian. We are also interested in epigenetic diversity in the neurons, and specifically, we study demethylation in the mouse brain using single nucleus methylome sequencing that we developed in the lab. In addition, we want to understand this epigenetic signature in the context of other features of the neuron, such as the neuronal connectivity. To study this, I developed a method called epi-retro-seq that combines both the retrograde tracing with single nucleus methylome sequencing. So first, we want to generate a comprehensive map of the demethylation profile in the mouse brain. We use single nucleus methylome sequencing and acid more than 110,000 cells from 45 different brain regions, including part of isocortex, olfactory area, hippocampus, and cerebral nuclei. Those are the same samples that were used for the single nucleus ataxic data Sebastian just talked about. So we have very nice matching data set. With this data set, we performed 3-level iterative clustering analysis and predicted 161 subtypes as laid out here. As you can see, we see this really diverse methylation profiles across different cell types, similar to the diversity revealed by the single-cell RNA study and ataxic study. What we are most excited about this data set is that the ability to directly predict enhancers from this data set. So we actually combined both our methylation data and the single-cell ataxic data from Bing Ren's lab and predicted 1.6 enhancer-like regulatory elements. Here I'm showing on the right some examples of enhancers predicted around this gene BCL11B, both upstream and downstream. So this gene is specifically hypomethylated in CA1, indicating an active state. And the enhancers predicted around this gene are also hypomethylated in CA1, indicating the active state of the enhancer element. So could this be a case of active enhancer regulating this active transcription of the gene in a very CA1 specific fashion? To further analyze this, we use a method called SNM3C-seq, a method developed in the lab to combine the chromatin conformation capture and the methylation sequencing. Using this method in hippocampus, we visualize the chromatin contact space in different hippocampus cell types. As you can see on the left, each cell type has a unique chromatin conformation signature. One of the examples is visualized on the right and highlighted in blue. So this chromatin loop interaction between two chromatin regions is specific in CA1 but not present in DG. In this chromatin loop, the interacting partners are about 500 kb apart. On one end, it contains a promoter region of a specific FOXP1 transcript, and the promoter region is hypomethylated with strong ataxic signal in CA1. On the other end, the interacting enhancers are also hypomethylated and with strong ataxic signal also in CA1. So this is a nice example of active enhancer interacting with promoter in a cell type specific fashion. And this is only one example of almost 14,000 CA1 enhancers at a specifically associated with CA1 specific chromatin loops. So now we have a comprehensive map of the regulatory landscape in the mouse brain neurons. What we want to understand further is how this epigenetic diversity correlates with the neuronal connectivity. In our study, we use epi-retro-seq and label the projection neurons with GFP by injecting AV retro in one of the 10 projection targets. And from each of the injections, we dissected out up to 8 cortical sources. From each dissection, we isolate the GFP-labeled projection neurons using flow cytometry and analyze them using single nucleus mesosome sequencing. At the end, we gathered data from almost 12,000 projection neurons. And for each of them, we have 3 information: cell location, the source, where they project to the target, and their methylation profile. Using all the information together, we can start to understand how different factors contribute to the demethylation variation between neurons. In this case, both the source region, the cell body location, and the projection target contribute to the methylation variation in projection neurons, but to different degrees. To take a closer look at this, we focused on the layer 5 ET neurons and further analyzed the heterogeneity within layer 5 ET neurons and identified 15 subclusters. Comparing the subclusters to where the neuron locates the source, it's clear to us that the source alone does not explain the heterogeneity. For example, for the MOP neurons, the red neurons on the map, they locate in multiple subclusters, suggesting other factors are in play defining the heterogeneity. So whether that could be the projection targets, to investigate that, we further take all the MOP neurons, identified subclusters within MOP neurons, and then overlay that information with the projection target information of neurons. So repeating this analysis for all the neurons from all the source region to different projection targets, we have this comprehensive map that from which we can right away see something really interesting. For example, for the modular projecting neurons from MOP or SSP, we can see on the corner those modular projecting neurons are clearly enriched in specific subclusters in MOP or SSP. This suggests that this specific subcluster in MOP or SSP, a defining feature is modular projection. And those modular projecting neurons have distinct demethylation profiles from other layer 5 ET neurons. In addition, we have a very interesting observation that some of the neurons projecting to primary visual cortex from ACE or RSP co-cluster with the layer 5 ET neuron. So this is really interesting to us that it supports there could be a rare population of layer 5 ET neuron that also project to cortical targets. So we verified the existence of those neurons by labeling the cell body of RSP neurons that project to primary visual cortex. Therefore, we can examine the axonal projections, and indeed, we see the clear axonal projection signals in thalamus, superior colliculars, and pons, supporting that those neurons are indeed simultaneously projecting to ET targets and primary visual cortex. So this is one nice example of the power of our data and the epiretro-seq, that not only can we study the epigenetic signatures of different distinct projection neurons, we can also identify interesting rare populations. So with that, I'd like to thank every member of our really fantastic Samba group led by Joe and Ed. So our next speaker is Yoram Mokomo from UCSD. He'll be talking about the joint analysis of epigenetic data and transcriptomic data.
Okay, well, thank you for the opportunity to present our work here, and thanks to the organizers of this really fascinating symposium. I'm excited to share with you the work of a really large collaboration. This has been a collaborative effort of a working group as part of the BICCN to investigate joint epigenomic and transcriptomic analyses of the data sets, specifically focusing on the mouse primary motor cortex. So as you've seen from the previous speakers, the BICCN is really making amazing strides towards building an annotated parts list for the brain. And you know, we take the perspective that having this parts list and understanding the molecular identities and something about the connectivity and other features of the cellular components of the brain is really the first step towards understanding function. But a question arises with the data sets, which is how do we arrive at some kind of validity for the level of resolution at which we divide the components of the brain. And this was really recognized in the Brain Initiative's founding documents, where they identified, identifying cell types as the starting point for intellectual technical progress. Just to contrast, artificial neural networks that have been so successful in computation use, in general, a very limited set of cell types, and we're really interested in understanding how the diversity of biological cell types contributes to brain circuit function. So, as I mentioned, we focused, as a group, we decided within the BICCN to focus on the primary motor cortex as an initial test case, and we produced data sets including over 600,000 high-quality single cells from a variety of technologies and platforms. So we had transcriptomic data, some of which has been shown by the previous speakers, epigenomic data, including open chromatin from ATAC-seq and DNA methylation from SNMC-seq that you heard about from Juju and Sebastian previous to me. And so this plot shows the number of cells that we have from each modality, and I think it's instructive to look at the trade-off between the number of cells here on the x-axis that can be profiled with each modality. This is really a function of the cost of the techniques versus the amount of information that's obtained per cell, which scales with the number of unique molecules that are captured and sequenced. And so you can see on one end of the spectrum we have techniques like ATAC-seq and 10x-based RNA-seq that profile many cells with fewer reads per cell, compared with the epigenomic data from DNA methylation on the left that shows much higher information per cell for a more modest number of cells. So one of the first questions we asked was what cell types do we derive when we analyze each of these data sets, each of the modalities on their own. And so in terms of transcriptomic data, you can see here, this is already a joint analysis of 7 data sets. So these are 7 different transcriptome data sets. And you can see the expected diversity of excitatory and inhibitory cell types as well as glial cells. We combine this with meta-neighbor analysis. This is work performed by Jesse Gillis's lab using their technique that looks at the statistical replicability of individual cell types across data sets. So it's really a stringent test of how well a given cell type can be predicted based on signatures that are present in totally independent data sets. And the result was that over 70 transcriptomic cell types could be reliably replicated with a high replicability score. We found similar patterns using the DNA methylation data on its own or the ATAC-seq data on its own. And the next step was really to look into integrating these data sets. So here you can see examples of the signatures of a particular gene, KLK2A, in terms of DNA methylation, DNA accessibility, and gene expression. And the motivation for integrating them is really threefold. So first of all, by integrating at the single cell level, we're able to validate the predicted cell types across these really independent biological features. The integrated data set also gives us molecular signatures of brain cell types across modalities, and ultimately we're interested in understanding mechanisms of gene regulation and persistent cell type identity, and that really requires comparing at the single cell level the epigenetic and transcriptomic regulation. So we treated this problem as a missing data problem, wherein for each individual cell that we profiled, we have one set of features, for example, gene expression, but we're missing information about the complementary features such as DNA methylation and chromatin accessibility. We took 2 different approaches to filling in these missing data. Single cell fusion developed in my lab, and Liger that you heard about earlier from Evan McCosco and Josh Welch. And in both cases used the gene body signatures of regulation, so gene-body chromatin accessibility or gene-body DNA methylation, as well as the gene expression level estimated from the RNA-seq data, as a common ground for integrating the cell types. So we're able to link cells across data sets by using these gene body signatures. And following the integration, we can then go back and look at intergenic distal elements such as enhancers that were not used directly for the integration. So the joint analysis identified over 50 cell types. We actually came up with 56 cell types using both single cell fusion and Liger. You can see that the cell types integrate very well across all of the modalities. And just again to emphasize, this includes both transcriptomic and epigenomic information. So it's really, it was very encouraging to see this high level of integration. Moreover, the cell types that were derived from the integrated data were highly consistent with those that came from the RNA-seq analysis on its own. The number of cells from each modality was also consistent across cell types. One really, I think, striking way of visualizing the consistency of these data sets is to look at particular genes. So for example, this gene TSHC2 has a very clear signature of gene expression as well as open chromatin and low DNA methylation in the same group of cell types. And so here, these heat maps show similar plots for I think 30 or so marker genes, and you can see really a high level of conservation across the cell types. Again, once we have the integrated cell types, we could then create browser views where we look at the level of individual cell types, the DNA methylation, open chromatin, and gene expression, not just at gene bodies, but also in distal elements. And so I just want to advertise that this is a resource that's available we actually have multiple resources that have been shared in our bioRxiv preprint, including this genome browser, as well as other tools for analyzing individual cells. Okay, so how can we address this question of validating the cell types and the resolution of cell types? Well, at one level, we saw that at a very coarse-grained level of cell types, we have very consistent signatures of the cell types across all of the modalities. Zooming into just the MGE-derived GABAergic neurons, we see that subtypes, individual subclasses of parvalbumin or SST cells, also have very consistent signatures across each of the modalities that were included in this joint data set. The meta-neighbor analysis also could be refined to the level of individual cell types, and so this plot is showing the area under the ROC curve derived from meta-neighbor, which again, the red blocks along the diagonal illustrate that we have very strong, very high replicability of cell types across each of the modalities. And this is focusing specifically on the transcriptomic data set. We went on to develop a couple of different ways of looking at cross-validated measure of the quality of the clustering. And so to do this, we split both the genomic features by dividing the chromosomes into two sets, training and testing, as well as dividing cells into training and testing sets. And by doing this, we could derive a cross-validated measure of the mean squared error of our cluster predictions. And we could do this as a function of the resolution of the clusters. So by varying a resolution parameter, we could find a U-shaped curve that shows the minimum mean squared error at a resolution of around 60 to 100 cell types, in this case for the SMART-seq RNA-seq data set. We performed this same cross-validation analysis across each of the 8 data sets and derived the optimal number of clusters. A more stringent type of cross-validation was performed across data sets, where we actually, again, divided the gene sets into training and testing, and then performed the integration analysis using just training data, and then used that to derive an optimal number of clusters. And we did this also as a function of the number of cells. So by downsampling the data sets, we could look at how the number of clusters that we're able to estimate scales with the size of the data set. And interestingly, we found that as the size of the data sets approaches several tens of thousands or more, we do see some signs of saturation of the number of cross-validated clusters across data sets. So just to acknowledge the folks who contributed to this work, this was really, as I mentioned, a large collaboration of over 80 collaborators. The lead authors on the joint analysis were Yong Yao, Hanxing Liu, Fengming Shi, and Stefan Fischer, and Hongqi Zheng at the Allen Institute, and I were the leaders of the overall working group. And we just want to thank the funders in the BRAIN Initiative, as well as all of our collaborators within SEMBA, the Epigenomic Consortium. And with that, I'd like to go ahead and turn it over to Basilka Tasic from the Allen Institute.
Thank you very much for organizing this meeting and for inviting me to give a talk. Today I will talk about the viral genetic tools for targeting mouse and primate brain cell types. And I would really like to start with acknowledging that I'm very fortunate to be part of the Allen Institute, where team science is encouraged and supported. And what I will present today is the result of efforts of several scientific and support, as well as superb leadership within the cell types program by the Allen leaders Hongqi Zheng and Ed Lean. I would also like to highlight several colleagues who have played a major role in this project: Lucas Graybuck and Tanya Daigle from Mouse Genetic Tools team, and John Mitch Boaz-Levy and Jonathan Ting, who lead the Human Genetic Tools and the newly formed CNS Gene Therapy team. We have also collaborated with teams outside the Allen and have received generous support from the BRAIN Initiative. We have had a long history of building and sharing genetic tools with the science community. This effort at the Allen was spearheaded by Hongqi Zheng and is now led by Tanya Daigle and me. All Allen mice go to JAX, where they're broadly distributed to the scientific community. And what I would like to highlight in today's talk is that there is a game-changing approach in genetic tools that relies not on germline transgenesis, which will certainly continue to be important, but on adeno-associated virus vectors that enable cell class or cell type specific expression in the mouse brain. The work I will highlight today was first published in bioRxiv in 2019, and we updated it 2 publications this year. It is currently in revision. These are Graybuck and Daigle et al. And Mish et al. And I will be able to provide only highlights today, but for more details, please refer to those 2 preprints. So how did this change in the way we design and build genetic tools come to be? It was really enabled by new single-cell genomic profiling technologies that allow us to measure thousands of genetic features in a cell, transcripts or open chromatin sites, and then use those features to systematically classify cells. This classification not only provides hierarchical relationships of cells, from broad categories like classes to finer like subclasses and types, but it also reveals specific genes, many of them previously not known as marker genes, as well as the putative regulatory elements in the case of a TAC-seq that can be used to make genetic tools. And here I'm just highlighting some of the genes, some of the markers that are specifically expressed in certain classes, subclasses, or types. These technologies also reveal cell type diversity and homology across species, here exemplified by 2 taxonomies, one from mouse and the other from human cortex. And having this information really begs the question, can we make tools that work across species? To do this, because germline transmission is either not ethically or practically feasible in many organisms besides mouse, we focused our effort on finding putative regulatory elements that could be isolated from the genome, cloned into adeno-associated virus vectors and delivered to animals without germline transmission. To define these elements, we use single-cell attack-seq. The two data sets are described in the two preprints, one from mouse visual cortex and the other from human middle temporal gyrus. Without going into details, I would like to highlight that differential chromatin accessibility can be used to define cell clusters, mostly at the level of subclass. We can correlate these data with single-cell transcriptomics, and then we can identify putative regulatory elements that are specific to certain attack-seq clusters. Then we can take these differentially accessible regions, some that are present in one class or one type versus all others, and use them as putative enhancers to build new genetic tools. And our workflow is on the left. So what we do is we first prioritize the cell subclass or type that we want to focus on. Then we find genomic regions that are specifically accessible, and sometimes we also look at proximity of marker genes and the conservation. We clone these into recombinant adeno-associated virus genomes. These either drive directly fluorophores or exogenous recombinases. Then we produce and screen these viruses, and there is really a variety of screening platforms we employ, but many of them rely initially on the mouse. And then for the most promising candidates, we actually test them across species using either in vivo or in vitro approaches. I will be able to highlight just a couple of tools. The first one I would like to highlight focused on layer 5 subcortically projecting neurons, also known as layer 5 pyramidal tract or extra-telencephalic neurons. We selected a number of putative enhancers and built genetic tools that drive either directly a fluorophore or a recombinase. We observed a pretty remarkable specificity with a direct fluorophore and recombinase. Roughly about 90% of the cells labeled were layer 5 PT cells. I would also like to highlight that the completeness of labeling depends on the route of administration of the reagent. In this particular case, in retroorbital delivery in mice, the completeness is relatively low, only about 30%. However, it can be improved with direct stereotaxic injection, as I will highlight on the next slide. We're also able to examine the expression of these enhancer viruses throughout the whole brain using 2-photon tomography with tissue side, and have also shown that some of these tools can work in human brain sizes to label equivalent cell subclass. In the next slide, I will highlight putative. Tools built by focusing on the parvalbumin interneuron subclass. In this case, we tested 3 different, I'm just highlighting the 3 different enhancers that were tested by human genetic tools. In fact, this particular experiment was performed by performing stereotaxic injections in the macaque brain. 3 different viruses were tested and subsequently assayed for specificity and completeness of labeling them immunohistochemistry. And these 3 different constructs containing 3 different enhancers really displayed excellent specificity at 86 to 95% and good to quite excellent completeness at 63 to 92%. I would also like to mention that these tools in mice are compatible with standard mouse transgenes. And in this particular case, we've built a new tool that allows genetically different populations of cells to be labeled in a single animal. So if you deliver 3 different enhancers cloned into 3 different viruses into this new tool, AI213, delivered through retroorbital injections, we can observe in the same piece of tissue 2 different layer 5 excitatory neurons, excitatory subclasses of neurons, and dispersed throughout the cortex inhibitory neurons. But these tools are also compatible with existing tools and novel tools like the one highlighted here. I will briefly just highlight that we have now together as a team effort at the Allen, been able to cover truly most major classes of neurons. So on the right, I'm showing as part of the mouse cortical taxonomy, the coverage by viruses that we have produced at the Allen. In blue are the ones validated by MFISH or single-cell RNA-seq by specificity. And in gray are the ones labeled or validated by just preliminary observation of cell location or morphology. And I'm happy to report that we have coverage of all major classes of neurons, as well as some non-neuronal classes, and are making inroads to covering finer divisions in the taxonomy, like specific types of paralbumin cells, for example, chandeliers. Where do we go from here? I would be really excited to collaborate across BICCN to integrate data sets and analysis, and therefore expand this approach to other extracortical brain areas. Then we would, in a coordinated fashion, proceed to build and screen AAV tools and characterize the most promising one in multiple platforms and species. And ultimately, what I hope to provide, we will provide to the community are publicly available data sets, reagents, and recipes for targeting cell classes and subclasses in the mouse and primate brain. Take transgene X and virus Y to be able to label cell type Z. And with this, I would like to complete this session. I would like to thank our founder, Paul Allen, for his vision, encouragement, and support. Thank you for your attention, and I would like to open the session for questions.
Okay, I would like to thank you, all the speakers. And now the podium is open for submitting questions. So for the audience who want to submit questions, please click Ask a Question button and send you the question in. So the first question, molecular omics data may reflect some axis of biological identity, such as developmental specification based on genetically based patterning. We know neurons differ with respect to many observable phenotypic features, including morphology, location, connectivity, electrophysiological properties, besides genetic makeup and transcriptomic characteristics. Moreover, neurons are highly influenced by a large number of interacting cells. Can transcriptomic and epigenomic profiles of brain cells become a surrogate and a sufficient definition of the cell types? So maybe start with Evan.
Yeah, sure. This is a great question. It's one that I think a lot of us here on the panel think a lot about. I like to think of this question. I mean, I think the answer is no, not until we really understand the relationship between the transcriptomic features and these other phenotypic features. And I think the best analogy that I've gotten to this problem is from human genetics. So in human genetics, you can imagine clustering people based on their overall, genotype, right? And that genotype is going to be most heavily influenced by ancestry and things like that. But if you really wanted to understand all the features of a person, it would be impossible to use those clusters to do that. So, for example, lactose intolerance, right, which is a phenotype that some people care a lot about. That phenotype is driven by, one or just a couple genes. And so you would only really be able to relate that to the genome if you understood the relationship between that phenotype and that genotype. Just clustering based on the overall, features is not going to give you that insight, because people are not going to, there's so many other influences besides that one, lactose influence that's affecting the genotype. And so you're only going to get the overall broad brushstrokes. So it's really about, next era is to really find relationships between these other features that we know are so important with the transcriptomic features. And I think that's what you've seen in many of these presentations today. Basilka, you also mentioned you might want to answer this too.
Yeah, no, I completely agree, and I think this is really a wonderful analogy. I just want to say that, beyond, there are a couple of things that we need to take into account. First, the measurements and the quality of measurements of different modalities really differs. Therefore, we may sometimes conclude that there is no, let's say, difference in one modality or that there is in another. But actually, maybe when we're able to measure more features with higher accuracy, something that appears either as a no difference or as a continuum may in fact become discrete in that modality that maybe we couldn't measure very well. And the second thing is I completely agree with you, understanding relationships really matters, because when you measure everything, some fine relationships may be, some relationships that depend on very few features may be hidden. So,
okay, thank you.
Can I maybe jump in and comment on this question too? Because I think it's a great question.
Okay, come on.
Is that okay? This is Aran Mukana. I completely agree with what Evan and Basilka said, and we really need to understand how these things hang together. At the same time, I think one of the lessons, one of the remarkable things to me of this field is how correlated many of the phenotypes of cells are. And so, as Juju showed, for example, in her talk, there didn't necessarily have to be a transcriptomic or a DNA methylation signature of the connectivity pattern of neurons. But it turns out that there is, and we can infer certain aspects of the projection pattern of cortical projection neurons from the DNA methylation. And I think we're still at the very early days of understanding all of those relationships. But I think our experience with the data so far, I would say, gives hope that there are going to be very robust signatures that are correlated across many of these modalities that will allow us to use the molecular signatures as kind of a defining core classification. But that's a hypothesis that I think we're all testing.
Hi, this is Juju. I just want to quickly chime in that I agree completely with you, Ron, that we are just scratching the surface of all the relationships from our data. I think it will be very exciting to see with more data in the future and more modalities, what kind of connections or relationships between those different features. And also, I think with the epigenetic data we also have the hope to understand the regulation underlying those connections that, for example, enhancers from the talk you heard about, whether we can use enhancers to really label certain cell types with certain specific properties and further study them.
Okay, the next related question is really about the consistency of the molecular characterization. So this is to Iran's talk. The mini-FS study indicates that mouse primary motor cortex contains over 55 neuronal cell types that are highly replicable across molecular analysis methods. However, computational data integration of single cell and single nucleus RNA-seq data alone can enable clustering and identification of 90 neuronal types in the same region. Could you please clarify what this difference in the number of neuronal types is due to the relatively low number of cells sampled in epigenomic profiling modalities, or some due to some different statistical criteria used for clustering?
Yes, thank you for the question. This question was really the reason that we wanted to so carefully look at objective measures of cross-validation of the cell types to be able to address this question of what's the right level of resolution. We talk a lot about lumpers and splitters, and two different groups or two different scientists can look at the same data set and come to different conclusions about what the right grouping of cell types may be. So I would say actually the results of our analysis were quite consistent. So actually, if you look at the number of replicable clusters that we found, for example, using MetaNeighbor in the RNA-seq data was somewhere around 70. The 55 neuronal cell types you're referring to from the epigenomic data, that's actually from joint analysis of the epigenomic and transcriptomic data, which actually included all of the cells, so over 600,000 cells. So that certainly wasn't limited by the number of cells sampled. But one thing I do want to emphasize is that, you know, we report kind of the minimum mean squared error, the point at which the clustering that has the minimum mean squared error, but there was really a range of cluster resolutions that was compatible with the data, and which had roughly similar kind of agreement with the data. And so I think what that tells us is that there's not, at least these data don't clearly define whether there are exactly 55 cell types or 56 or 65. There's a range, and you know, that range spans, you know, from something like 50 to 100 cell types. You know, it's far more than I think many of us would have anticipated, a few years ago. But it's not a single number, and so I think our challenge is now to describe what those cell types are and how do they relate to each other going forward.
Any other speakers?
Hello, this is Sebastian. I'm also thinking that the cell cluster, the cell number, is a very difficult question from that point of view, that some might be different based on a transcriptome, others might be different based on epigenomic data. So I think at this point it's really hard to understand until we have really phenotypic electric circuit data to really understand what are the distinct cell types even doing, or are these kind of like molecular phenotypes that we don't understand fully.
Okay, so I received several specific questions, and one is for Sebastian from Dr. Gregory Horowitz at the University of Washington. What fraction of putative enhancers were associated with the nearest gene on the linear genome?
Yeah, thank you for that question. We have not looked into the fraction of the nearest gene. I would presume it's somewhere around like 50%, but we haven't looked in that particular data set.
So the next question is for Bosilka from Dr. Koichi Hashikawa at the University of Washington. Is the viral approaches to target cell types, can you drive the expression of CRE or FRIP under the enhancer elements while ensuring specificity?
Sorry, I'm not sure I understand. There is also, I already answered how to control the specificity. I think it was also by Koichi Hashikawa. Is that sort of an equivalent kind of question? How to control specificity with viruses, given that there are so many aspects that you have to control? Yong, do you think that's the same question?
Yeah, so he thinks the small leak of CRE and FRIP may potentially reduce the specificity dramatically.
Yes, so I think viruses, though more convenient, more easily shareable, more portable, yes, there are many challenges. One is titer. With titer, things will vary. With method of delivery, the specificity and completeness will vary. Whether it's a recombinase or it's a direct 4-4, again, the specificity and completeness will vary. So I think in order for viruses to really be reliably used, we have to provide recipes. Virus, this exact virus with this titer, delivered through this method at this dose gives this particular specificity. Yes, I think with transgenes is easier, especially if it's a direct fluorophore transgene. I mean, there may be some differences with genetic background of the level of expression or maybe specificity, but very minor. Here, it's much bigger. So I think that's what we have to do. We have to test, provide recipes for people to be able to use in the field. And with recombinases, we have indeed noticed that some recombinases do decrease specificity, like CRE. But if you take CRE that is not as active, actually it approaches a very good specificity of FLIP. So we've done some tests internally.
Okay, I have one general question from Dr. Sanyarski at Salk Institute. What was the rationale for focusing on the motor cortex? What is consistency in cell classes and subclasses across all the groups studying cell identity? So I might want to chime in as a program director of this Brain Initiative Cell Census Network. And so in my introduction slides, we currently have more than 20 projects in this large consortium, and each project is going to generate a different set of cell census data. As you heard today, so we actually selected molecular characterization groups to report, as we have already collected a large amount of data, more than 10 million single-cell omics data already released to the public. And at the same time, we have another major group characterizing cell features, from the angle of anatomical and imaging approaches. And these methods, the throughput is relatively low, and so the data generation is a bit, at this moment, is a bit lagging behind. But there are a lot of interesting data already generated and will be reported soon as well. And the focus on the motor cortex is because this is a relatively well-known region, and contains about 2 million cells in mouse brain. And so we want to start this focused region, and so we can compare different technologies and protocols so that the data generated will be better integrated together. And so especially, when many technologies are relatively new, and so the standards need to be developed during this data generation process. So that's a major reason, a rationale. And any other panelists want to chime in on that?
This is Trygve. Just wanted to say briefly, I think I mentioned in my talk, but it also was very fortuitous for a comparative analysis, because this is a functionally conserved area and was also identifiable in all species with some high confidence. So that's the only other thing I wanted to add.
Yeah, and this is Aran. Just to add to, I think Yong and Trygve mentioned the main kind of motivations for choosing MOP. In addition, it's an interesting part of cortex that has, shares most of the kind of cytoarchitecture with other cortical regions in terms of the layers and the cell types, but it also has some differences in terms of being an agranular cortex, and there were some open questions about, the status of layer 4, for example, that we were able to address with some new data. So I think that made it interesting for us as well.
So here's another question. So one hypothesis that neuronal connectivity, functional properties, and disease states may be encoded by cellular transcriptome and the epigenome. Do you have data suggest that other observable phenotypic features may indeed be inferred by the analysis of single-cell omics data and by multi-modal assays? And this may be also related to another question for Zhu Zhang about ET neurons by Sue Custer at NMH.
Thank you for the question. So yeah, I think from our data, we can clearly see that using at least the demethylation, in many cases, we could build a prediction model to infer the projection targets of the neuron based on their the location of the neuron. And I want to highlight that this prediction really varies in different situations. For some of the projections, for example, the medulla example I showed, it's really quite clear cut. But for some other projections, for example, the pons versus superior colliculars, that difference really depending on which brain region, which cortical region we are looking at. I think we look into that. That really comes from many of the neurons have dual projection, where our study only look at one single projection target. So I think earlier we talked about this. I think it's like there's a lot more to be learned. I think we're really just starting to see how much information is encoded in the epigenome or transcriptome. And for the layer 5 ET neurons that project to cortical targets. So in this case, we look at a zero cortex projection, and in anterior cingulate ACA, we analyze more than 1,000 neurons, and about 30 of them projected to, more than 1,000 layer 5 ET neurons, more than 30 projected to zero cortex, so about 3%. In RSP, the percentage is a bit higher, about 10%. So out of about 500 cells, layer 5 ET cells, we saw about 50 neurons that project to zero cortex. Thanks.
Yong, I think there was also a part of that question about, if I understood correctly, about whether we saw evidence of cell state differences or activity dependence of the either epigenomic or transcriptomic elements. And just to address that, I mean, I think overall the BICCN has really been focused on, characterizing the landscape of stable cell type identities. And so we've been very careful to focus on, sampling animals in uniform conditions, and haven't focused on activity dependence. But I think having a baseline knowledge of the landscape of all of the cell types and will allow us going forward to look at that. I think we do expect to find, for example, activity regulated differences in enhancer activity and gene expression. But that isn't something that we've directly looked at. I don't know if others want to comment on that as well.
Well, this is Trygve. I just wanted to comment actually on the original question of sort of linking omics profiles to other cellular features. And I mean, I think, as you said, we're just now collecting the data to start to asking these questions about creating predictive models. But I just wanted to highlight too that the comparative analysis defining these homologous types across species actually is a predictive model of sorts, in that, for example, chandelier cells, right, we already knew that the morphologies looked quite similar across species, but we went into this with an unbiased approach and identified that as sort of a one-to-one match. And so, in fact, could have predicted the morphology of chandelier cells in primates based on the morphology in mouse. And of course, there'll be species specializations over evolution, but at least it gives a starting point for making predictions of phenotypes, particularly for phenotypes that can't easily or really at all be measured in, for example, human brains, such as long-range projections. So we have also homologous types for example, the corticospinal neurons, and that would be very difficult, if not impossible, to sort of really verify in a human brain. So I think that's just another use of the comparative approach.
Yeah, and I know we are close to out of time, but just jumping on Trygve and Aaron's last comments, one thing that this next phase is particularly poised to do is really think about inter-individual variability and thinking about more diverse genomic and genetic backgrounds. But the corollary of that is really getting a handle on the environmental influence on transcriptomes. So one thing that we have observed is that we see high inter-individual variability across human samples, but much less so amongst laboratory-reared non-human primates, which are just as genetically diverse. And so that really puts an emphasis on understanding how much environmental variability we might need to face when we move to either wild type or human samples.
So here's probably a quick question to the panel members. Is your study already published? Maybe starting with Evan.
Oh, ours is on bioRxiv. I think a lot of the studies are part of our, the recent compendium of BICCN papers, and we're all quite committed to releasing them on bioRxiv in advance.
Yeah, so I just want to mention the most presentations discussed today, if not published in bioRxiv, and will be submitted to Nature or bioRxiv soon. And so this is an ongoing effort right now for the Brain Initiative Cell Sensor Network investigators to publish their results and also try to integrate data together moving forward. So with that, I think our time is almost up, and I want to just ask each speakers if they have any quick one-sentence summary or about their work or some future direction they want to take. Bosilka, maybe.
I was thinking, is somebody going to speak before me? No. Okay. I'm really, really excited about what we were able to do within BICCN. It's really amazing. Working together, generating all these data, and then analyzing them together to find common rules and common types across different modalities. Again, mentioning that different data do not always give the same answer, but there is really an amazing agreement. So my hope is really that we can leverage these data to build genetic tools. For me, this would be, I think, transformative for the whole neuroscience field, and not only in mouse, but also across species. I've just received a couple of emails about rat, primate. I think that would be really a game changer in non-genetic animals. And sorry, that was more than one sentence.
Just to chime in on that, this is Trygve. I just wanted to say, as we're studying this sort of regulatory genome and understanding and identifying, for example, viral enhancers, I think we have an opportunity to really study cell type evolution and principles of that as we understand that regulatory logic across species. And I also think this sort of, again, looking at different species, and particularly model organisms, can really maybe guide good selection of those model organisms for the community at large. So I look forward to future coordination of focusing on individual brain regions, and sort of the collaborative analysis effort, I think, was really fruitful.
All right, I'll chime in. Our cerebellum study, I think, really surprised us that that novel cell types can be discovered even in the modern era in a structure that's been so heavily studied. I think the sort of relatively untouched structures of the hindbrain, the midbrain, and other subcortical structures, I think, are going to provide the most immediate and obvious use to the neuroscience community as we just discover very basic principles of tissue structure that we didn't know before.
So this is around, I think my one sentence would be, that some people sometimes ask the question, is it too many cell types? There was a paper with that title by Chuck Stevens from 20 years ago that I came across. And I think what this has this collaboration has shown me is that the data really insists to us that there are, a very a broad diversity of types in the brain. And I think the next challenge for us is to understand why and what they're doing, and also how they arise in development. So I think we have some of the data that can help us to understand them from that perspective as well.
This is Juju. Just want to echo everything you've already said, but I think from our study, it's really just amazing to us to see how complicated the whole system is. All the different factors could contribute to basically all project to the variation we see in the demethylation or other modalities. So I think it's really, like we've already said a lot, like really important to connect all the different modalities. And also the enhancer identification and validation functional study is definitely a future direction we are very interested in and actively pursuing.
Anything to add, Sebastian and Fenner?
Yeah, just another synthesis of what's already been said, but what we've come to appreciate is measuring more than one species, more than one region certainly gave us insights that we would not otherwise have had, both in the ways that cell types evolve and also how they might develop differently in different genetic contexts. So development, I think, is a clear new path that BICCN is already invested in, and that could lead to real insights into how some of these novelties or unexpected cell types or states come to be.
And this is Sebastian. So what I think is it really shows, and what really excites me is kind of like to have this integrated approach between all the molecular modalities and the phenotype and the anatomy, and really trying to understand. And I think now we might be on the verge to be able to understand how gene regulation and enhancer elements can kind of like contribute, and where is it encoded in the genome, this diversity. So I'm really excited about what will come out of this comprehensive analysis that we will conduct in the future.
Okay, with that, I want to wrap up this symposium by thanking again all symposium speakers and the audience as well. Thank you.
Thank you.
Thank you.
Thank you.
Thanks.